Hello, my name is Seth Crosby, and I am speaking to you from St. Louis, Missouri. I'm at Washington University. My contact information is at the very bottom of this screen, and any of you that have any questions or would like to access the technology I'm going to talk about today, you're most welcome to write. I'd like to thank LabRoots for the invitation to give this talk. Today I'm going to be talking about an unusual way that we have invented to characterize microbiome samples using multiple 16S hypervariable regions. This was invented by our director, Richard Head, and our bioinformatician, Andrew Schriffer, who has since then moved on. He's no longer with us. Another co-inventor is one that I'm sure that many of you will recognize. Jeff Gordon is also an author on the paper, which is going to be referenced during this presentation. You're most welcome to read it if you like. All right. So today I'm going to talk for about 1 minute about our facility, and then I'm going to give maybe another minute or two on microbiome profiling, sort of general comments, and then we'll dive into talking about 16S sequencing, why it's useful. And finally, most of the talk is going to be dedicated to MVRSION, which is a method that we have used to characterize 16S. And I'll be talking about a road test we took with it, where we were comparing it with a very much used protocol called QIIME. And much of that, again, is captured in the paper that you're welcome to look at. So a moment about us. I started a facility about 8 or 9 years ago called the Genome Technology Access Center. It was a CAP/CLIA certified sequencing facility that was for smaller projects. The McDonnell Genome Institute has been a resident at Washington University for decades. And it was there that much of the Human Genome Project was done, as well as many other genomes. And recently we've combined into a single facility. So we have tried to keep the best of both of our qualities, and you can learn more about that. We're so new right now. We're still working on the website and working on our rebranding and all that kind of stuff, but happy to talk about that offline with anyone who's interested. This is just a slide my boss likes me to show, just showing that during and through this process, both of our teams have been publishing lots of papers or helping with the publication of lots of papers since the inception. And we both occupy buildings, so we're all going to be moving over from Cortex into that chocolate-covered building. So we'll be physically together in a few months. When I was in medical school, when we thought about the critters that are either on us or inside of us, the one way that we had for characterizing those was by culturing them. And one thing we knew is that many of the bacteria that are on us and in us are not culturable. So we could only get sort of a moderate snapshot of what's going on there. With the advent of high throughput next generation sequencing, we started to be able to, rather than culture out of a specimen, we could actually sequence out of a specimen. And this tends to be done in two different ways. One common way is shotgun sequencing. And this is where you grind up all the DNA in a specimen and make it into libraries and sequence everything that's there. So you'll get representation of the entire bacterial genome. It can be quite accurate. It's a little bit expensive because you have to sequence more deeply in order to be able to get a good representation of what's there. And it can be a little bit dirtier because not only are you grinding up often the bacteria that's present in a specimen, but you're also grinding up the host DNA as well, which is often not as informative. So one way to address this problem is rather than sequence the entire bacterial genome, scientists a number of years ago began sequencing just one of the genes. It was the gene that coded for one of the ribosomes in bacteria, the 16S ribosome. This is a picture of the protein of the RNA and the protein bundled together. This is the RNA in 16S. And you can see that, like all RNA, it assumes a secondary structure of a lot of stems and a lot of loops. This tends to be relatively similar. The secondary structure is similar from species to species. And the way that's done is that these stems tend to be relatively conserved from species to species. These loops, however, are quite variable. In fact, they're referred to as hypervariable between species. And these can be species specific, so that if you sequence these loops, you might be able to you're able to tell what species the DNA came from. And even though we're talking about ribosomal RNA, as this is what it codes for, we are sequencing the DNA, not the RNA. We're sequencing the gene that codes for the ribosomal RNA. So what you have is you have a bunch of, these are the stems in blue and the loops in yellow. And sorry, I've been using my cursor up until now. I forgot that doesn't work here. So I'm going to be a little bit more verbal now. The nice thing you can do here is that you can take advantage of those conserved regions to design primers to do amplicon PCR. So you design primers for the blue domain. Those tend to work over lots and lots of species, and then you sequence across the yellow domain, which is species specific. Traditionally, the way this is done is you concentrate on one hypervariable region. It would be quite difficult to be able to try and compare all the hypervariable regions at once. That would result in 9 different lists, and you wouldn't know which list was the true one and which one is not. So people will tend to pick out the hypervariable region that they think is most informative, and we'll do that. And so here we can see we're amplifying region V4, sequencing it, and then taking that into bioinformatic analysis. Most often people use a program called QIIME, which I'll talk about later, to do that bioinformatic analysis. What I'm going to be talking about today, what we're doing with MVRSION, is we're doing sort of what I alluded to in the last slide, is we're trying to amplify up all of the hypervariable regions. And not only are we, you can see here, not only are we amplifying up all the hypervariable regions, but in some cases, those conserved regions aren't quite as conserved as we hoped they would be. And so we will actually design multiple primers for individual conserved regions to try and get as much of all of the hypervariable space as we can. There's one slight modification that's happened recently to this, is we no longer use V9. We don't find that it is particularly helpful in terms of calling species level information. So you can see there in the little table that I've drawn there, that I've drawn a line through V9. Another thing you'll notice is that the amplicon size never exceeds 300. And the reason is that when we do the sequencing, we do paired end 2 by 150. So the most we're able to see is 300. And so we hold the amplicons down to 300 bases. We use a high throughput PCR method from a company called Fluidigm. This allows us to do thousands of PCR reactions in a single run. It makes our job much easier, and it also reflects in a lower cost of the assay. Then we do next generation sequencing, and the bioinformatic analysis we're using now is no longer QIIME, but it's MVRSION. And there you can see a reference to the paper the first paper that we published about this. Subsequently, users have begun using this, and so I believe it's also alluded to in subsequent publications. We find that by using all of the hypervariable regions rather than just 1 or 2, that we're able to get more accurate calls, more species level calls rather than genus level calls, and less false positives. And I'll talk about that when I present a little bit of the data that we generated. So we're going to be looking at the positive MVRSION and the sensitivity of MVRSION. And sometimes there are students in the audience who will get bored with my talks. And so I try to make the talk a little bit more interesting, at the same time to remind them of what positive predictive value and sensitivity is. And I don't think that any of the talks that I've seen on LabRoots actually involve any games. So it's time we're going to play a game. Very simple game is in the next slide, I'm going to show you a series of circles. I'd like you to count the circles, all the circles that are on the slide, except for those little green circles we're looking at. They just gave me a new template and that contains circles. You don't need to count those, but some circles will very briefly appear on the screen. I'd like you to count them and then just sort of, normally I have people raise their hands, but just count the number and then have it in mind and we'll see how close you can get to the right number. So watch the screen because it's not going to be on for more than a second. Okay. Okay. How many circles do you think that you were able to see on the screen just back then? All right. So here's the answer. There are 6 circles. 2 of them are not exactly circles. Those are false positives. And in microbiome work, a false positive is when you identify a genus or a species of a bacteria which is not actually present in your sample. And false positives, by the way, is one way that in this study, MVRSION really stands out. And you'll see that in a little bit. All right, so that's positive predictive value. Is the higher the positive predictive value, the lower the number of false positives. Now let's find out how sensitive everyone in the audience is. So I'm going to do exactly the same thing. Just count the circles that you see on the screen, and we'll be back in a second. Here goes. Okay, how many circles did you get? And so often when I give this, people tend to be looking in that rectangle, but there's actually circles that were there that you didn't identify. And these are false negatives. In a microbiome sample, these would be species of bacteria that are present in the sample that weren't detected by your assay. And this is a false negative. So the positive predictive value measures the extent of false positives in a sample. The more false positives, which is in the denominator, the lower the positive predictive value is. And sensitivity measures false negatives. The more false negatives in a sample, the lower the sensitivity. So in the case of using amplicon sequencing for microbiome work, the cause of false positives can, especially in the case when you're using a single hypervariable region rather than all of them, is sometimes, even though I've told you that the hypervariable region can be different from species to species, sometimes, especially within a genus, the hypervariable region can be so similar that the caller will mistake the species or not be able to identify the species and will provide a genus rather than a species. And these are false positives where you're actually detecting either the wrong thing or not exactly what you're looking for. A genus isn't exactly a false positive, but often people who do this work would rather have species level call rather than genus level call. For anyone who's done PCR, the cause of false negatives is relatively obvious. It's that sometimes the primers don't anneal. We've already talked about the fact that some conserved region primers won't anneal to some conserved regions. Sometimes the resulting amplicon, there's so few bacteria that are present that it's a too low concentration and it doesn't show up in the sequence. This is very important. If a hypervariable region is not present in your underlying database in how you analyze all the hypervariable regions, it's not going to be detected. And there are other reasons why using amplicon-based sequencing can sometimes result in a false negative, which is the bacteria is present in the sample, but you're not detecting it. So I'm going to talk now about right there where all the arrows are pointing toward that little plastic cassette. As I said, it's made by a company called Fluidigm. And here's sort of a closer shot of it. And what you do is in this case, it's got 48 wells on one side and 48 wells on the other. You load your samples onto the left side and you load your primer pairs onto the right side. And then using microfluidics the samples and primers are mixed in 30 nanoliter chambers. I don't remember how many there are, but there's 48 by 48 reactions that are occurring in just an hour or two. And then once these reactions have finished, they go into a single chamber where they can be withdrawn, and they're sequence ready. They're ready to go onto the HiSeq or onto the MiSeq. Depending on the number of samples you're using, that's how we decide what Illumina instrument to use. More recently, Fluidigm came up with another system called the Juno. In this case, the number of primer pairs you have is reduced to 24, which is more than enough for us. But what you get in exchange for that is you can run 192 samples at the same time. When we're running this many samples at one time and then sequencing them all in a single run, the cost per sample can get to be quite low. All right, so we decided to test this thing, and what we did, you can see in the center there, the red and the yellow, is we used the simultaneous use of 8 regions using MVRSION, and we compared that against a gold standard, which is QIIME, and that is how QIIME is spelled. And in this case, we used 2 regions, regions 3 and regions 4. One thing I want to say about how we configured QIIME is we wanted this to be an apples-to-apples experiment. We knew that by sampling 8 regions that the MVRSION is extremely sensitive. So what we did was we opened up the QIIME thresholds all the way so the QIIME sensitivity would be almost as good as the MVRSION sensitivity. So what you'll see in the data when I present it is that the sensitivity is not very different, but what happens is that the specificity, the positive predictive value, is quite different, and you'll see that in some of the data that I show you. What we fed into this experiment were 3 different defined sets. One we got from the government, the BEI set, and this is 20 equimolar. It's a cocktail of 20 DNAs from 20 different species in an equimolar concentration. In addition, one of our co-authors, Jeff Gordon, provided us some defined 48 sample sets. And in this case, they were equimolar, but also he gave us the same bacteria in staggered concentrations. The concentrations of these 48 genomes varied over 4 orders of magnitude. And the reason he gave us that is we wanted to find out 2 things from MVRSION. We wanted to figure out how specific and sensitive it was. We also wanted to know how well it is at predicting the abundance of bacteria. So by varying the abundance, this will help us with that problem. In addition, we got some real-world sets as well. These were completely defined samples from germ-free mice, which have been fed certain previously identified bacteria. And I believe that there's some samples from human patients as well. So we ran these through QIIME, through MVRSION, and we looked for call accuracy, sensitivity, and positive predictive value. And also we looked at the abundance accuracy. And there again is the reference to the paper if you'd like to look that up. So first, what I'd like to do is I'd like to explain, before I get to the data itself, I'd like to explain as best I can. I am not a computational biologist, but I'll try and do a good job. And by the way, if you find that my explanation leaves you still hungry for more, please contact me and I can put you in touch with the inventor, and he can give you a lot more precise detail than I can about how MVRSION works. It's kind of a complicated protocol. The idea, though, is to be able to simultaneously, in parallel, look at all 8 hypervariable regions and use them to be able to make species level call in your data. These slides are available, I believe, if you need them, so please don't try and write down what I'm saying. So what we have here is we have sort of different going along at the same time. And those 3 parallel arrows represent alignments. The one parallel arrow is L-shaped upside down. It's there on the right side, but the alignments happen 3 times, and I'll talk about those as I go along. But I'm going to go ahead and stick within my red circle, and that red circle is going to move around the screen, and I'm going to talk about how this works. So I'm going to talk first about the in silico side of what we do. This requires no actual lab work at all. This is all being done computationally. And the first step is a very important step. This mVersion protocol does not work unless it has a highly curated database. So we take all of the available, we start with a public database, take all of that data, and then we basically get rid of the aspects of that database which are not useful. So, for example, we get rid of sequences. In the top blue, you can see sequences with genus, sequences with strain but no species, sequences with inconsistent characterizations, fungus, viruses, all that stuff we try to remove. And so, for example, we started, if in that olive-colored box, we started with the Silva release, and we had about 600,000 16S genes in that. And after cleaning it up, we ended up with 125 species-specific 16S sequences. And I told you something that's not quite right. I said we're getting rid of the annotations that have only genus level. I asked them actually to bring the genus level back in. And so what we do is we run the protocol, we make all the species level calls that we can make, and then we take what's left, any unidentified amplicons, and if we can, we go ahead and run the program again, this time using the genus level. So we do a good job at identifying species, but we try to leave no stone unturned, and we also do genus level calls on the things that we can't get species off of. So this is just a picture of the curation of our database. So what we do next, now we have in that little cylinder that says curated 16S database, those are full-length 16S genes in that list. Next, what we do is we determine which hypervariable regions might be amplified on our lab bench. And so we do in silico PCR. We basically, on the computer, we run PCR using a program called PCR Prospector, and we determine which hypervariable regions should get amplified. And this goes down to that cylinder called all predicted amplicon sequences. So now we don't have full-length 16S anymore. Now we have a bunch of predicted amplicons. And what we've done is we filtered away amplicons which should not appear in the data. The whole thing that we're doing with mVersion is that we're constantly trying to filter away things which shouldn't be there, based on different parameters. So okay, so now we're going to stop on the in silico side, and we're going to move over to the bench side. So as I said before, we do 2x150 paired end sequencing. We take whole DNA from our whatever biospecimen we have. We add all those primers, and we end up with a whole bunch of hypervariable region amplicons, hypervariable region 1 through 8. And then what we'll do is we align all of those amplicons to the curated full-length database. We're not using our predicted amplicons yet. We're still up at the top. You can see that first horizontal arrow is to the full-length database. And what this does is this filters away any species which should not be there. And we end up with a database called candidate species. So these are amplicons from species which aligned to our highly curated database. What we have in our candidate species is a whole lot of false positives. We're only interested in the gene in getting rid of the species which should not be there based on our database. So now we have a list of species that are in our sample, and we have a list of species that might be in our sample. So we need to further refine this list. So now what we do is we take that list, and this is sort of the most complicated aspect of MVersion, is we align all of the predicted amplicons that we got from in silico PCR with all the amplicons of this species that might be in our samples. And what we're looking at is we're looking for amplicons which align specifically to the smallest number of species that they can. So, for example, in species A, an amplicon hypervariable region number 1 might align rather promiscuously in the data that we generated on our Illumina instrument. But hypervariable region number 2 might align very specifically. So for species A, we're going to say, okay, hypervariable region number 2 is our best hypervariable region. We're going to kind of ignore hypervariable region number 1. However, in species B, it could be that hypervariable region number 1 is the one that aligns most specifically, whereas hypervariable region number 2 is a little bit more promiscuous. So we go through all of the predicted amplicons and we hybridize them to all of the species that might be in our data in order to determine which predicted amplicon is the most valuable for any particular species. And this is how we're able to use all 8 hypervariable regions simultaneously, is that we weight the hypervariable regions using only the hypervariable region which is most informative for any particular species. So now we take all of those, the most valuable of the predicted amplicons, and we do yet another alignment. We go back to the original data and we align the highly weighted hypervariable regions to the original data, and this allows us to make the final species call and also to be able to predict abundance. So I'm going to show you just a little bit of data. There's much more in the paper if you're interested. So in the top 3 scatter plots, what we have here is a comparison between the predicted abundance, because in all of our defined samples, we knew how much of the bacteria should be in that sample, and on the x-axis is the actual measured, the actual amount that we observed in the samples. So MVersion is on the left, and then we have the hypervariable region number 3 of wide open QIIME, very hypersensitive QIIME, and region 4, a wide open QIIME. And what you can see at first is that for a very abundant species, MVersion and QIIME tend to work relatively well, about as good as the other. The data doesn't look very different. The predictions and the measurements look relatively close. However, as you get into less abundant species, this is where QIIME becomes less informative. And naturally so, because you're only using a single hypervariable region. If you're using all 8 hypervariable regions, picking the best hypervariable region, the most informative one, you maintain sensitivity all the way down across all those orders of magnitude. In the bar graphs below, what you see is each triplet of bars is the left one is MVersion, the middle one is QIIME, and the right one is QIIME. So, and what we're comparing is the first 3 bars are true positives, the next 3 bars are false positives, and the next 3 bars are false negatives. So the false negatives show us what I already told you, is that the sensitivity, when you open QIIME up all the way, the QIIME tends to be about as sensitive as MVersion. It was able to detect almost the same number of species as MVersion did. However, because the sensitivity is so high in QIIME, as most of you know, when you increase sensitivity, you decrease specificity. The false positive calls were completely different in QIIME as compared to MVersion. MVRSION is very sensitive to use all 8 hypervariable regions. It's also quite specific as well. All right, so that is the end of my talk concerning this new tool called MVRSION, which increases the accuracy of species level calls. I want to continue to reiterate that this is not genus, this is species. We can see that the abundance estimates are accurate down to very low abundance. I want to reiterate that the sort of one of the aspects, the secret sauce of MVRSION is the fact that we use a highly curated database. The MVRSION does not work well unless the database is curated. And I also want to reiterate the fact that because we're doing these things in such a high throughput manner, the library prep and the sequencing can go down to as low as around $30 a sample, which we think is very useful for the research community. Future directions, we'd like to continue to incorporate other, we have Silva now, we'd like to bring in other public databases to make sure that our underlying database is as complete as possible. And we would like to bring in other primers as well. We're missing some species as indicated in the data that I showed you. We don't get 100% of the species, and this may be that there are sticky places in the conserved regions, and we're going to design more primers to try and cover those. In addition, we'd like to be able to do eukaryotic calls as well. We'd like to be able to detect various fungal species, and so this we'll be using the ITS 1 and 2 genes to do that. And I know that all the questions that you have are in the future, when this is finally broadcast. But I welcome them. I believe that LabRoots is going to provide you a means to communicate with me. And this is a very old slide. I just yesterday delivered that young lady to the University of Missouri at Columbia, where she's now a freshman. So I probably need to get a new slide. Thank you very much for your time.